So we'll look at very briefly a number of points. The situation that pertained prior to 2007, because that's a bit of a watershed in this subject. How big a shock the initial findings of ENCODE were to the biological community. We'll try and understand what is now defined as the epigenome. What does it actually mean? And then in the final part of the talk, to see the implications both for Darwinism and also for design. I'm sorry, very sorry that Steve is not here. Never mind, doesn't matter. Another day that changed the course of biology was February the 16th, 2001, when in two prestigious journals on the same day, Nature and Science, the draft of the human genome was published. In fact, President Clinton and Mr. Blair and lots of other people were on the news saying that this has completely revolutionized the way we see life. We've decoded the book of life. Actually, it was just the beginning of a journey. It wasn't the end of a journey at all. What we discovered, some of these things we probably appreciated before the final draft or the draft of the human genome was published, was that within each cell, within our 23 pairs of chromosomes, we have approximately 3 billion letters of DNA. A lot of material. One of the shocks really was that at that time, at first count, it appeared that we only had 22,000 or so, give or take a bit, because wherever you look you see a slightly different number of genes. This wasn't anticipated because of the concept that we perhaps are as complex as anything else there is in terms of living creatures or organisms. We should have very many more genes, and as Ard quite rightly pointed out, we have about the same number of genes as a nematode worm. And so this is a bit of a shock. It makes us more humble, probably. I'm not sure whether that's true or not. And of course, when we add up the number of protein coding genes that there are in this enormous genome that we have, again, the numbers vary a little bit depending on what you read. It's between about 2 or 3% of the DNA is actually coding for proteins. These, in my opinion, are the nuts and bolts. It is really not at all surprising that we shared a very similar gene to the chimpanzee when we consider something like hemoglobin. Hemoglobin is a molecular machine. It does a very specific job. It's going to be very similar in many animals that have hemoglobin, that's for sure, including chimpanzees and gorillas and orangutans. It's not really surprising. If I just want to make one point about Ard's talk, is that this thought of 98 or 99% comparability between humans and chimps is a deception, because it's essentially based initially on protein structures. And we're talking of this 2 or 3% of the genome, or even protein structure itself. When you consider what we're going to consider now, we see very clear demarcation lines between species. And so whatever we thought of in days gone by, maybe before the year 2000 or certainly 2007, we need to revise our estimates on comparability, particularly if we want to use that as an argument for common descent. Now, the interesting thing is, I'm expanding this more than I should because of time, is that this is the talk that I gave in 2010. We looked at the evidence for common ancestry. We considered some of the things that Ard also put forward today as evidence for common descent. There are counter-arguments, and there are more counter-arguments today to that form of reasoning than at any time in history, and it is just going to get worse, as we will see later on. Of course, we could say this, since only 2 or 3% of our DNA codes for proteins, it is true that certainly prior to 2007 and years before that, it was quite often that we would speak, or people would speak, of the rest of the DNA actually being junk. Nobody talks in those terms anymore, although there's a backlash, and I'll come to that at the end of the talk. The thought is that all of this extraneous material can be in part just a relic of an evolutionary past. I think it's true that ENCODE has changed that. What does ENCODE mean? It's the encyclopedia of DNA elements. And if you can see the highlighted letters, you see how they come up with the word ENCODE. The first publication was on the 14th of June 2007. This is an incredible date for me personally. And I'll tell you why. I'd been invited to come and speak at ELF in Eger, and that conference began on the 16th of June 2007. I was in Budapest, staying with my wife in a flat. Would there be any possibility that I could get internet access? Actually, not only could I get internet access, but I could download this paper that was published in the journal Nature in its entirety, which is an amazing thought when there's such a restriction from getting academic papers, particularly from these very prestigious academic journals. In my flat in Budapest, the days before I came to Eger, I was made aware of this amazing development. And of course, I was able to put it into my talk that I gave. Really, I want to give honour to the Lord for this, because although I don't, I'm just an ordinary guy who wants to keep up with the biology, I nevertheless feel that when I want to give such technical talks as I would give here, that the Lord really enables me to do it by, look at this, Geoff, this is important. This is what the paper said: identification and analysis of functional elements in 1% of the human genome by the ENCODE pilot project. The thing that is so startling about this work, and it is only the 1%, is that what was discovered, that the vast majority, they didn't put a figure on it in those days, the vast majority of the genome is pervasively transcribed. That means the DNA is transcribed and you get RNA sequences off of that DNA template. This was not expected. There was initial skepticism, and there continues to be skepticism today. And we'll look at some of that skepticism towards the end. Of course, September of last year, 2010, the full-blown work was published, an integrated encyclopedia of DNA elements in the human genome. And just to highlight some specific words, how did they ascribe function? Well, they looked at transcription, transcription factor association, chromatin structure, and histone modification. And beyond that, by looking at all of these features, they were able to assign biochemical functions to around 80% of the genome. This is very controversial, but we'll listen to the words of the authors. Can you hear that okay? It's not too loud, is it? Move a bit round it. So what is the epigenome? It's an interesting title. Well, I think the first thing we can say is it's definitely a higher level of encrypted information. And it is involved in the regulation of those 2 or 3% of the genome which are the protein coding genes. In other words, the 80% or maybe the 77% is involved in the regulation of the expression of the 2 or 3%. And the fact is, new papers are published in this area every day. I mean, this is the most incredible, explosive information moment in the history of biology, probably, because every day in the academic literature there's a new paper which is involved in helping us to understand better epigenetics and the epigenome. And so actually it's a little bit, we're very much at the beginning of this journey, one would have to say. So to talk about it now, maybe, I'll be very old then, but maybe 2013 or 14, if biology comes round again, we can have another talk, an update on what we understand. But we know that it's all about the control of gene expression. And there are a number of areas to look at briefly: chromatin modification, non-protein coding RNAs, which is a bit of a mouthful. I never would want to call them non-coding RNAs because they're coding for something, that's the point. And so I've thought up a term. You heard it here first: epigenetic RNAs. And we need to briefly look at things like alternative splicing, because that came up in the talk yesterday with Siegfried. We need to consider enhancers and inhibitors of different types, which are all involved in regulating gene expression. And we definitely need to consider something about cellular differentiation, which is so incredible. So chromatin modification. I don't think there's any human engineering that can compete with this, Pete. Would you agree? Amazing. The unit is the nucleosome. Okay, so you've got histone proteins, which are very, very positively charged, around which you have wound a very negatively charged DNA. The phosphate, sugar phosphate backbone of the DNA means that it's negatively charged. The nucleosome, does it form, is it self-assembly? To a certain extent it must be true. The information creates these proteins and the structure, of course, of DNA, and there is a natural association between a very positively charged protein and a very negatively charged nucleic acid. And you see it here, the nucleosome, different way of looking at it: positively charged histone protein, negatively charged DNA. And you see that there's a histone tail which sticks out. You actually could see it on that animation sequence. Now we can modify that histone tail by adding acetyl groups. For example, if you add an acetyl group to a positively charged protein, it will become less positively charged, and therefore the strength of binding is weaker between DNA and the protein. Also, methyl groups can reduce that strength of binding. If you remove imino groups, you also get this activation of expression. In other words, the opening up of this incredible structure. On the other hand, if we remove the acetyl groups, or if we remove the methyl groups, or add imino groups, you get this strong reassociation of the negatively charged DNA with the histone protein. In addition to this, we need to consider that cytosine in the DNA sequence, one of the nucleotide bases, can be methylated. Actually, it can also be hydroxylated, but we'll consider methylation at this point. And you can see how these two things are highly coordinated. DNA and histone modification is a highly coordinated process, and it involves numerous enzymes, cofactors and proteins for it to work properly, basically. We'll see it in the clip. The HDAC is the enzyme, which is a deacetylase, which removes the acetyl groups from the histone. Okay, that's just a very quick overview of chromatin modification. This is part of epigenetics. This is one of the ways genes are expression is controlled. Moving on to epigenetic RNAs, they come from a whole host of different sources. If the DNA is pervasively transcribed, that is an indication that these epigenetic RNAs are coming from lots of different places within the genome, interestingly both from the sense and the antisense strands. Also coming from introns. We'll talk about introns a bit more. Also coming from pseudogenes. Oh dear, I heard that word a bit earlier. What's in a name? I believe, and I say this quite openly, that the term pseudogene is a misnomer. And because we have defined a pseudogene as not having a function, and you think I'm making this up, you need to read even current academic literature. Pseudogene has no function? What nonsense. Pseudogenes are actively involved in the control of gene expression, and I believe with all my heart, however we disagree over vitellogenin or vitamin C enzymes, that actually this is a prediction, okay? I'm predicting something on the basis of design, okay? I believe that one of the ways a pseudogene might appear in the genome is that something that was once functional for a very brief moment in time, and this is particularly true in embryonic development, may need to be switched off permanently, and there may be natural frameshift mutations or other types of mutation that's got nothing to do with random process, but is actually part of the control of the overall function of the genome. And that's a prediction. I wrote about vitamin C, I wrote about vitellogenin, and all right, I'm not in a position to do the experiments. The experiments are not being done because of the theories, because of this concept that vitamin C, gulo, which is the final enzyme in the sequence, is a pseudogene. It proves common ancestry. We don't want to know if in early embryonic life it actually is functional. And I believe with all my heart that it is functional in early embryonic life. And I have the papers to prove it. And I've written about it. And I've been castigated for writing about it by people that you know perfectly well. But the experiments, as Ard and I and Siegfried might think, they're not difficult experiments to do to show that even in embryonic stem cells, and we'll talk about that a bit later, one could look to see if there is the expression of those genes. Oh, if I could do that work, if only I had my own laboratory or research group, I would love to do experiments like that, because it would tell us what? That the use of pseudogenes as evidence of common ancestry is actually wrong? No! It would tell us much more about biology and this overarching wonderful control that there is in gene expression. And we'd have to redefine the term pseudogene. Here's a paper, briefly, that was published in February 2013. It's just one. There's lots of pseudogenes, okay? Pseudogene express non-coding RNA. It's not non-coding though. It's coding for something very important. It's got information in it. Interestingly, coming from the antisense strand of the pseudogene DNA. It's recently published paper. I could talk about this for ages. Mobile transposable elements, including retroviral inserts, as they're called. I dispute that, by the way, completely. And I've written about it as well, the counter-arguments. All right, here's a fantastic paper that I spoke about in 2010. I haven't got it written on a piece of paper. If you want it, or maybe I can give the PDF of this presentation to anybody who wants it. Look at this, incredible. Retroviral promoters, mobile promoters that can move across the genome, get associated with the gene sequence and cause the promotion of that sequence, the expression of that gene. Incredible. Here's a very recent paper. Transposable elements, no more junk DNA. I love this paper because it completely blows the whole idea of common ancestry out of the water. These things are species specific. Of course, Dennis Alexander can put one example in his book. Maybe I shouldn't say names in this talk. One example of commonality. But there are myriads of examples where there is total difference. And of course, transposable elements are no longer considered junk DNA. What are the types of epigenetic RNAs? There's short RNAs, there are long RNAs. I don't like the word non-coding. I put it in this slide. I don't like it because they're coding for something. Most recently, circular RNAs. What do? They block the continuing translation of messenger RNA. So in this you see the activities to block the continuing translation of messenger RNA. This is essential, that messenger RNA has a very short half-life. The amount of protein that is synthesized needs to be very carefully regulated. But there are also RNAs that block the blockers, and they act as molecular sponges. I mean, I'm not making up these words. Here's a paper, February 2013. Circular RNAs throw genetics for a loop. Actually, this is a comment on a paper in Nature. It's not the paper itself. Behold the latest curio in the cabinet of RNA oddities. Naturally occurring circular RNA molecules that influence gene expression. At least some of the loops, described in 2 papers published this week by Nature, act as molecular sponges, binding to and blocking tiny gene modulators called microRNAs. So these are the sponges that are blocking the blockers of mRNA translation. But the researchers suspect that the circular RNAs have many other functions. The molecules comprise a hidden parallel universe of unexplored RNAs. You know, we're right at the very beginning of this journey. Epigenic RNAs have specialist roles, and it's definitely worth mentioning this one, X inactivation. It's a really interesting phenomenon. It only happens in the female that has 2 X chromosomes. One of the chromosomes, interestingly, needs to be switched off. Very interesting phenomenon. And it does it by the binding of these long chain RNAs called exist. I suppose you could say zist. I'm not really sure how you pronounce that. But it binds all around the Y chromosome, and you get this compacted structure that is essentially shut down. An interesting phenomenon, why it is that the female embryo, if you like, which has 2 sex chromosomes, one of them has to be shut down. Interesting finding. So they're blockers, sponges, mimics and decoys. I think this is true certainly from RNA that's coming from pseudogenes. By definition, they can't produce a protein. Well, they do produce proteins in certain situations, but most often they are not going to. But they then act as a decoy. They're not going to produce the protein, and they will take up perhaps the ribosome and get it to produce nothing that's very useful. There are enhancers and inhibitors. All of this, inevitably, essentially, is under coordinated, exquisite control. Let's very quickly cover, I think I'm okay for time, something else which you might not read about so much in current popular books on epigenetics, of which there are 1 or 2, and I mentioned them in my handout. We need to go back to alternative splicing, because this is a phenomenon. What do we mean by that? Well, we have a protein coding gene which forms a long messenger RNA which contains introns and exons. And of course there are enzymes, or machinery actually, enzymes is a very good word here, machinery that cut out the introns and splice. I've made a mistake here, somebody pointed that out to me already once before. They don't slice, they splice exons together, and you get a processed messenger RNA which is used to go to the ribosome for translation and the formation of proteins. But there's alternative splicing, which will produce different types of protein, different variants of the same protein maybe, or something completely different. And so when we consider how many genes there are, all right, 22,000 or whatever, how many proteins there are, Well, it's very difficult to know, because alternative splicing will lead to more than one gene product from one gene sequence. This was exciting for me because the second time I was invited to speak here, which was in 2010, where we definitely looked at the issue of common ancestry in the talk, actually really interesting to note that there was another very significant paper published in the days leading up to that conference, which was called Deciphering, I spelled it right, the Splicing Code. Just to highlight a few words in the abstract, or I think the introduction: hundreds of RNA features, tissue-dependent changes, alternative splicing for thousands of exons, remember exons and introns, distinct regulatory programs, different tissues, mutation-verified regulatory sequences. Mutation puts a spanner in the works of this. I showed a fantastic animation of the spliceosome in my talk in 2010. I haven't got time to do it here, except to point out that it is probably the most complex molecular, macromolecular machine in the cell. The whole business of splicing and how it's affected and how it alters, it's just, it blows your mind. Also, going back to more classical days, biochemistry, we need to recognize that there are protein enhancers and inhibitors. I love this diagram. And you've got the coding region. This is the promoter region. You get binding, obviously, of the RNA polymerase. You've got all of these other proteins. You've got the tartar binding protein going to what is known as the tartar box. You've got all of these other proteins associated with it. And then, of course, you've got activator molecules, which could also be proteins. They will bind to specific sequences which are upstream. They're not part of the coding region. They are upstream of the coding region. You've got spaces within that sequence for repression, and things like steroids, for example, small molecules can be involved in that inhibition of gene expression. Amazing. Self-assembly. Yeah, why not? I suppose it does self-assemble, but all of these proteins have information at the heart of it. They don't just occur randomly. Oh, I just highlighted these different parts. I didn't use it. Never mind. Yeah. What's the bottom line to that? Life is complicated. It's true. Life is very complicated. But you heard Ewan Bernie, and watch this space for what is going to come out of these hundreds of scientists that have been involved in the ENCODE work. One of the criticisms, it's amazing to me, one of the criticisms of the ENCODE project is that they used pluripotent cells as their starting point. Actually, they used some cancer cells as well, but pluripotent cells, where you get so much potential gene expression. This is a criticism. I can't understand the criticism at all, because we know that the vast majority of the genome is potentially transcriptionally active. That's what ENCODE has shown. But we also recognize that there may be more than 200 different cell types, all of which have the whole information content, give or take a bit, that is in an embryonic stem cell. But in that case, the vast majority of it is switched off, transcriptionally silent. Has to be like that. It has to be like that in order for those cells to differentiate. And the process of cell differentiation is highly ordered and complex. And just to talk briefly about embryonic stem cells, just to show the difference here, you've got the fertilized egg, which is dividing, 8-cell embryo, maybe even to 16 cells. These are called totipotent cells. Each of these cells, if they're removed from this fertilized egg and kept separate, will do this. You'll get identical twins, because each of these totipotent cells has the potential to create the living organism, in this case human. However, once there is differentiation, and we'll look at that again in a bit more detail in a moment, and you get trophoblast cells on the surface and inner cell mass on the inside, it's only the cells of that inner cell mass that have the potential to go in lots of different directions, but they can't do everything anymore. They can't be part of the trophoblast that is involved in placental formation. They will only lead either to neural cells, cardiac muscle, blood cells, whatever. And of course, we're learning about the signals that are necessary to provide these inner cell mass cells, embryonic stem cells, that will lead them in all of those different directions. I mean, it's just incredible. We're learning what signals we need to add. The networks are all there. They're not changing, but we're adding appropriate signals. These cells are the most exciting cells of all, in my opinion. Sperm and egg cells, so highly differentiated. I mean, when you think of this little creature, well, you might even think of him as a creature, but actually he's only got half the number of chromosomes because of meiosis. What a specialized cell this is. He, in his little head, has got a nucleus that contains 23 individual chromosomes, but has all the information, well, give or take a bit, for a human being. A very highly differentiated cell. And of course you've got, on the other hand, the egg, likewise, highly differentiated cell. Epigenetic signals all over the place, shutting down so many of the systems. But the miracle is this: at fertilization, in the main, all of these epigenetic signals are erased, the signatures are erased, and you have the restoration of totipotency from two cells that are so highly specialized. Now, there are exceptions to this, and it's a bit technical. I spoke about X inactivation, all right? That continues. But also, there's this phenomenon of imprinting. What's imprinting? Well, you can actually detect a paternal signature and a maternal signature that is carried through this process, and it's called imprinting. Certain genes that are being expressed in the embryo have their origin either from the sperm or the egg, and you can detect that because there's an imprinting that's gone on. That signature hasn't been lost. But in the main, the vast majority of epigenetic signatures are erased. You know, all of these methylations, all of these acetylations, removed, like that. It's amazing. The cell divides and divides, and then begins to differentiate. And you've got, yes, as I mentioned, trophoblast cells, you've got a cavity, you've got an inner cell mass. We've seen that already. And then, of course, as days go by, there's the phenomenon of implantation into the womb, and you get this incredible increase in differentiation that's going on. The question that is still unanswered is what signals cause the initial cells of the blastocyst to differentiate and cause them to polarize? Can we now even go beyond DNA and consider other signals that are involved in this differentiation process? Well, I'll just list a few here, and I'll put this in this morning actually because it came out of our discussions after Steve's talk yesterday afternoon. Signals beyond DNA, cell-to-cell communication. I think there is a code in sugars as well. You know, proteins can be coated in sugars, and they have a very interesting defined sequence, and I think there could be coded information even in carbohydrate structures. It's just a thought that I've had. Cytoskeletons are obviously going to be involved in the spatial location of cell components. So as things begin to develop, you can see processes happening where there's presumably information content, but it doesn't actually involve DNA at all. So in conclusion, the implications for Darwinism and the implications for design. Firstly, the implication of Darwinism. As, I forget her name now, the editor of Nature said that we have discovered that disease seems to originate by mutation in the non-coding, it's not non-coding, in the regions that don't code for proteins, in that the rest of that genomic space. This is where diseases, mutation in these regions lead to disease. This was one of the big disappointments, I think, of the Human Genome Project. There was so much expectation that once we'd identified the genes, we would understand what caused disease and we'd be able to correct that. As it turned out, it's much, much more complicated than that. And in fact, most often, of course there are exceptions, you know, haemophilia is caused by a mutation in the gene coding for factor VIII. But in the main, mutation within the epigenome, if you want to call it that, the non-protein coding genome, invariably will lead to disease. Natural selection is a true phenomenon, but I believe with tremendous confidence that natural selection doesn't generate variation, but it removes that which is deleterious. And it is there to maintain, in part, genomic integrity and stability. This is another hour or 2-hour talk, which I wouldn't mind giving at some point, because at the end of the day, the integrity of the genome, and this is why I dispute totally this whole idea of chromosomal fusion, the integrity of the genome is essential to maintain fertility. Sexual reproduction is the greatest challenge to Darwinism that there is. And if there's space one day in a program at ELF where I could spell that out in detail, as I have done once or twice, I'd be very happy to do it. I have to leave it at that. It's not the survival of the fittest. It is not the survival of the fittest. It is the survival of the fertile. I mean, it's obvious. The survival of the fertile. But that is totally incompatible, not just with Darwinism, but with any other evolutionary scheme that could possibly be imagined. They will be incompatible with the maintenance of fertility. I come out, my background is not as a physicist, as I've already said. My background is reproductive endocrinology. I believe in this I have a measure of understanding. The thing that is so exciting for me, as definitely not a Darwinist, is that Darwinists themselves actually understand the implications of ENCODE. And I've brought a few papers. They've only just been published. This is such an interesting paper, and you really should read it. You should get it. You can get it freely here. And I haven't got it written down, but again, if anybody wants the PDF, look at the title of this. This guy is ranting and raving about ENCODE. On the immortality of television sets. I mean, where have you seen such a paper like this published in an academic journal? Function in the human genome according to the evolution-free gospel of ENCODE. I mean, this is not coming from a creationist or an intelligent design theorist. This is coming out of academia. They understand the implications of ENCODE, if it could possibly be true. And definitely get a chance to read this. It's such an exciting thing to get into the mindset of a Darwinist. He is stuck in a worldview, absolutely stuck in a worldview, and he can't move outside of it. Evolution for biology apparently is a fact, which it isn't, but apparently it is. And if you hold to a Darwinian worldview, it's very, very difficult to accept that something might actually be challenging that idea. Oops, let's go back. What they're worrying about is the definition of function. You see, function for a Darwinist is conserved through the process of natural selection. And there's two ways of looking at function. And they really want to look at the way that they don't believe the writers of ENCODE have done. They challenge them because they've looked at causal effects and they want selected effects as the definition of function. Because if function is conserved by natural selection, the fact is 80% of the genome can't be functional. It can't be. And they say the definition that ENCODE uses for function is wrong. The other thing that it goes into, which is really interesting, is that they now want to differentiate between junk and garbage. Garbage, of course, is something that will have no future use at all, so you usually throw it in the bin. Something that's junk, well, you put it in the loft, you think, oh, maybe one day it'd be useful. They want to distinguish between the two things. Because evolution has to have at least garbage. You've got to have something that's not going to affect the function of the organism to work on. That's the point. So there's got to be at least garbage there. You know, if we're showing that there is no such thing as junk DNA, well, beyond that, there's got to be something that evolution can work on. Another paper, this is much more modestly written. Can ENCODE tell us how much junk DNA we carry in our genome? And they make a very interesting point. Is it right to equate function with biochemical activity? Because this is how they've identified, or at least assigned function, to 80% of the genome. They've seen particular types of biochemical activity, which of course we've talked a little bit about, and they say this: biochemical activity is very different from functionality in the light of evolution. Interesting. And then we have Paul Doolittle, who's written this, again very recent, April 2013, Is Junk DNA Bunk? A Critique of ENCODE. And without going into all the details, and again it's really worth reading the paper to get inside the mind of a Darwinist, he does a thought experiment, and he's very interested in what is known as the Cvalue Paradox, which is an interesting phenomenon because many creatures, such as a lungfish, for example, have very, very much more DNA than we do. Amphibians, some of them, can have very much more DNA than we do. And he would say that if it's true, organisms with genomes larger than our genome should be considered as more complex than we are. If it's true that 80% or 90% of the genomes in those creatures, those animals, are also functional. Of course, we don't know, but we need to keep an open mind about it. Bunk, or how do I define bunk? That's good, you know, I'm speaking English. Bunk is just nonsense, I suppose. Yes, that's true, that's true. See, I mean, there's a lot of emotion involved in these papers, as you can see. Well, a lungfish has got unbelievable amounts of DNA. My recommendation to Dr. Doolittle is that he talks to the animals. That's a joke. Did anybody get it? No, nobody got it. Well, we need to do ENCODE, in other words, on all of these other creatures. Of course, if there's very much more DNA, of course, it's even more complex. And who's going to do the work? That's the question. Who's going to do it? Because ENCODE is involved in the human situation 400, 500, 600, 700 scientists. If we want to do the onion, he talks about the onion, which has got a bigger genome than the human. Well, somebody's got to do the work. You can't say that they're not more complex or less complex. We don't know the answer to it. And of course, there may be structural components that are important in chromatin. It's not a good time to be a Darwinist. Really, every day the literature is coming up with questions that Darwin cannot answer. This is my belief. We're living in the last days. I mean, even Steve yesterday in his talk in the afternoon quoted molecular biologists, evolutionary biologists who openly state that we're living in a post-Darwinian world. Don't nail your colours to a Darwinian mast because it's in the process of collapsing under the things that we are discovering in biology today. And it's going to get worse, folks. That's not to say that there won't be attempts to come up with other secular ideas, but mutation, natural selection, forget it. Forget it. It doesn't work. The implications of design are pretty obvious. The biological information that I think is the important aspect, because we had a discussion about what do we mean by information. The information that we're discussing here is not material. It's code that is carried somewhere but has a function somewhere else. It's passed from one to another. Language is another expression of an intelligent mind trying to communicate something that in this case is quite difficult to another intelligent mind. I'm speaking in English. When you have a problem like bunk, how do you interpret it? We've got to translate it into something else that's understandable, but the information remains the same. It's not material, and there is no naturalistic explanation for the origin of biological information, because it's not in itself material. The question to conclude, have I got time? How many levels are there? That's the question for us now. Now it's interesting because I didn't develop this idea this morning, but I developed it at home in Israel when I was thinking about how to conclude the talk. How can we understand these levels of information? And maybe this isn't such a complete understanding or helpful understanding, but at least it fits in with something that Ard said today and yesterday in our discussion. I thought also that you could conceive of the genome as music. And in addition to the music, we need instruments. And not only do we need instruments, but we need musicians that will be orchestrated and that will work together in a coordinated manner, creating a symphony. But there's something that's needed. We need the tap of the baton to get these people to come to order. And the question that I want to ask is, if we thought of the genome, the epigenome, can we consider that there might be above that overarching information that could be called the hypergenome? There you are, I'm giving you new definitions. You could say that you heard it here first. Okay? I don't know. But, although I know that Ard mentioned this, genes can't be considered the blueprint of life. Of course that's true. The genome is a one-dimensional code in a sense. The epigenome might be considered as a four-dimensional code. Encrypted information that is expressed in different ways in different cells over time. It makes me think of this verse in the Bible. For you created my inmost being, you knit me together in my mother's womb. I praise you, because I am fearfully and wonderfully made. Your works are wonderful. I know that full well. If you've got nothing from this talk in terms of the technical stuff, I hope that you catch a glimpse of the amazing creativity of God. And I just want to close with a little video clip that I made, which shows this incredible development of the embryo in utero, which at the beginning needs yolk as food source, but very shortly doesn't need it, because the placenta has been produced, that's the point, and its nutrition will be obtained from the mother. Although dietary intake of vitamin C does not explain what happens. You know, I'm going back to this old thought again, sorry about that. Well, I want you to worship God as we conclude. This is a very good question, David, and I hope that if I repeat the question, how is it that a liver cell is so specialized and yet that information is passed on to the next generation? And the liver cell has to be, the process of that specialization involves the switching off of so much of the information that's in the DNA that is contained within the liver cell. Well, we don't need to think about the liver cell actually, because whatever happens to the liver cell, it's not going to be passed on to the generation. The important thought is the sperm cell, which is so highly differentiated, where again the vast majority of the genome is switched off, probably more so than in the liver cell, but of course other genes are switched on. And of course this sperm cell is such a highly specialist type of cell, but it contains in its head, if you like, in it the nucleus. Well, it's only one pair of each chromosome because meiosis has already occurred. But effectively, there are some qualifications to this, but effectively virtually all of the information content that's going to create a human being is to a certain extent in the sperm, to a certain extent in the egg, but of course when you've got fertilization and the reunification of 23 pairs of chromosomes, you've got all the information, and all of it is potentially transcribable. That is just a miracle, because you're moving from highly specialist cells, which are switched off largely because of epigenetic signaling, and all of a sudden that epigenetic restriction to gene expression is released at fertilization. And of course, within the fertilized egg, you've got all the information that ultimately will lead, not if it's got a Y chromosome in there, ultimately will lead to the production of another sperm cell through this incredible process of differentiation. I've not explained myself well enough. Oh, of course they divide mitotically. Of course that's true. And the daughter liver cells that have divided from the parent liver cell will have those epigenetic signatures associated with them. They will not be erased. It's only at fertilization that you get the erasure, not of everything, but the erasure of most of these epigenetic signaling. I wrote a chapter in our book, Andy's book and my book, of muntjacs, mice and men, where I look at exactly what you're speaking about, Siegfried, that there is chromosomal fusion, there is chromosomal division, which does occur. The muntjac is really incredible, because you've got the Indian muntjac and the African muntjac, I can't quite remember. One of them has, I think, something like 23 pairs of chromosomes. The other muntjac has, I think, about 4 pairs of chromosomes. I can't remember the exact numbers. There's this enormous fusion process that has occurred. The fact is, the muntjac and the muntjac are still a muntjac. They're almost indistinguishable phenotypically, and I think that this is also true in the mice. Yes, that it couldn't happen. Oh, no, I'm not saying that at all. No, I'm not saying that at all. Yes. Can I just add to that point, though? The maintenance of chromosomal integrity is absolutely vital in meiosis. And therefore, that's where reproductive isolation obviously occurs in the muntjac and possibly in the mouse. because you cannot get meiosis unless you've got very accurate, and that's why the centromeres and the telomeres and all of those thoughts about that become an essential prerequisite to consider when you're thinking of successful fertility. Darwin can't get around that problem, just can't. I mean, donkeys and horses can breed, they have different numbers of chromosomes. I don't believe you could say that the donkey is the result of either a fusion or a splitting process. I don't know because I haven't looked into it, but they cannot breed. The mule, as far as I understand it, is sterile. And that's the problem. Well, I agree completely with that point. I have an evolutionary biologist here, so I have to be very careful about what I say as a generalization. But there is such a thing as, is it mosaicism? Genetic mosaicism, which I think does explain that very small percentage of fertility even in the mule. I do know about Seafried, I agree.